All right, everybody, we're going to jump right into the Visual System Part 2 lecture. If you haven't watched Part 1, obviously you should do that now. These two work together, and that means that you're not really ready to watch Part 2 until you've watched Part 1. There are a lot of details in this case that are important. I'm also going to try and keep this a little shorter because there were a lot of introductory details for the last lecture. As a bit of a recap, let's just remind everyone, just to prime you for what we're going to be talking about, how visual information reaches the brain. So first, we have the optic nerve coming from the retina, and these form the bundle at the back of the eye. And we have information coming from the left and the right visual fields, and that information is going to travel to where information from the left visual field will go to the right lateral geniculate nucleus, and this information in the right lateral geniculate nucleus is then going to pass back to the right primary visual cortex, also called the striate cortex. And information from the right visual field will go to the left lateral geniculate nucleus, and it will pass to the left primary visual cortex. So we have some information from each eye crossing over and some staying on the same side, all depending on the visual field or the portion of the visual field that it's coming from. And again, this all comes back to primary visual cortex. This is kind of the final step in getting from the eye into the main processing in the brain. However, the last step beyond this is for it to go on to secondary visual cortex and higher order association areas. And you should remember we ended last class talking about different streams of information because there are quite a few places once we get to primary visual cortex that things start to branch off to. And so you can kind of see here the entire path if you follow from the left visual field to the right side of each eye, to the right lateral geniculate nucleus, and back to the primary visual cortex. So that's it. If you're still having trouble, kind of putting these pieces together, I would walk through these slowly because they're very important. But otherwise, you should use this as a flow diagram for what's happening. And it's worth knowing all of the different terms here. You might want to list primary visual cortex, striate, V1 altogether, and secondary visual cortex, V2, extra striate cortex, just to help you when you're going through the book and things like that. So now we finally get into how we process some of this information. How do we perceive things like light, color, contrast, movement? These are all things that we're going to talk about in today's lecture. In terms of detecting light-dark differences, which contribute to a lot of different things, for example, how we perceive contrast, how we detect the edges of things, or even just the ways that we perceive something being lighter versus darker in general. So this is where we need to go back to these on and off center cells. So looking quickly back at these on-center ganglion cells, they have the highest rate of firing when they have light precisely in the center of their receptive field. And you can see this here. However, there's kind of a second thing that can happen, whereas if they have light in what we call the surround area or the peripheral component of their receptive field, then they're inhibited. So it's not just that they fire at the same rate, but they're actually shut down by that. And this is going to become very important in a second. Whereas the off-center ganglion cells, they are inhibited if they have light falling on the center of their visual or receptive field, and they become excited by light that happens in kind of this peripheral or surround area, and that's when they have the highest firing rate. So let's put this into a practical example. I think this diagram or picture rather from the book does an incredible job of this, where if we're trying to detect the difference between where one object starts and another one stops, and there is some sort of a contrast difference, let's assume this is not something very bland, then what you're going to have are a bunch of these on-center ganglion cells, and they're going to have competition that's happening, because if this area is lighter, then of course we have a brighter light on center, but we also have this bright light hitting the edges too. And so these two kind of arithmetically combine, right? This means that we're going to have illumination that's causing them to fire more action potentials, but they're partially inhibited because that illumination also falls on the edges as well. So they're not firing at their maximum firing rate. However, you're going to have, at least at the precise edge between two things, you're going to have a set of neurons that are able to fire at their optimal firing rate, right? So these bipolar cells that are getting this different set of information, of course, are passing this on to the ganglion cells, who are the ones actually firing these action potentials. But these on-center cells that have the light on the center, but then also have something darker on the surround, that means that they are able to fire at close to, if not completely at that maximum firing rate, because they're not receiving this inhibition from light falling on the edge or the surround of their peripheral field. So keeping that in mind, I want you to notice something. So if you look at the squares up here, then what you'll notice is that the very edge of this lightest square, as you get towards the next square in line, appears a little bit lighter. And if you look from this side and we go over towards this edge, the actual finite edge here appears a little bit darker. And you can see this for each of the squares, right? So there's kind of a lighter edge on the right and a darker edge on the left. And this is actually a bit of a trick. The squares are exactly the same all the way across. And if I take them and I separate them, they look much more uniform than they do here. But this is just a screenshot of what is exactly sitting inside of this square. And this is really these bipolar cells playing this trick, where when you have something on falling on the edge, just like we have all of these edges here, our brain and our retina essentially amplify the edges by using this kind of on-cell, off-cell trick. Switching directions a little bit, it turns out that color vision works in a similar way, but it took us a long time to figure this out, mainly because people started to think about the ways that we perceive color. And for the longest time, there was an argument in the field of vision neuroscience about exactly how this would happen, mainly because there were two theories that could kind of explain what we might be able to do equally well. The first we call the trichromatic theory. And this theory came about because we know that, at least at the level of the retina, there are three different kinds of cells. We have cells that can detect cones, specifically, that can detect red, green, and blue. And this would mean that all the way through the visual system, we would have a red, green, and a blue cell, for example. Now, this ends up being pretty interesting because the main downfall of this theory is that it can't account for certain things. For example, for us, yellow is a primary color, and it's considered to be fairly pure, and yet we don't have cells that specifically detect yellow. So this is kind of an interesting thing where the brain perceives this as primary, but the retina doesn't have yellow cells. It's kind of a head scratcher. And the other thing that it really fails to account for is the way that we blend certain colors. So I know that many of you, if I asked you to, could picture a blue-green color. So do that now. Think about how the different ways that you can blend blue and green. Now, most of you are probably thinking of something that looks kind of like a turquoise, and you do this all the time. What if I ask you to tell me, or to think about, what a reddish green looks like? Think about that for a second. It's a little bit more difficult because we're not really able to blend those colors together. It's not something that we're used to seeing together. And in fact, if you think about mixing red and green, what you're going to end up with in your head is something that's brown and not really any sort of color that you can latch onto. So what this kind of suggests, if we think about the way that these colors come together, is that the visual system is designed more for what we would think of as an opponent color system, where there are certain colors that we're able to blend together, but that there are others that are sort of contrary to one another. So red versus green in that last example is something that we are not able to blend together because we have a system that detects those as opposites. kind of like we just saw for the example of black versus white on edges. So the idea was that an opponent system would be something like yellow versus blue, or red versus green. One of the problems, at least early on with this, is people couldn't figure out exactly how many different cell types that we would need to make all of the different combinations. Do we need opposing then red-yellow cells and green-blue cells? Or how exactly do we make this combination to get all of the different colors that we have? So this question went on for a long time, and like many things in science, it turned out the reason that it went on for a long time is that both sides were wrong, just a little bit wrong, not perfectly right in either case. And it turns out that our brain and our eye work together to use both of these systems. So it's not purely a trichromatic system, and it's not purely an opponent color system. So it turns out that within the retina, we have a trichromatic system. We have cones, specifically in the retina, that respond to either blue, red, or green. And these are incredibly specific to those colors. However, you'll notice that we don't have a yellow, right? So that there were some things about the problems with the trichromatic system that remain to be true. And the way that we're able to detect these different wavelengths in the cones is that we have specific opsins in each of the different cones that are specifically tuned to one color. So the blue cone has a set of opsins that are tuned to somewhere around 400 or 420 nanometers in terms of the wavelength of light. And then we also have green cones and red cones. And the rods, as if it were designed in some really clever kind of way, respond somewhere near the middle of all of these. And you might remember this is kind of the way that the opsins we looked at in chapter 5 for optogenetics were designed. There was one that responded to blue light and another that responded to a yellow or a green light. So these things are out in nature. They're out in the ocean animals where those opsins for controlling the brain were originally kind of taken from, and then they were changed into very specific proteins. But out in the world, we know that now there are many of these different opsins that exist, and they're just proteins that are light sensitive, in many cases sensitive to a specific wavelength of light. So I want you to take a second here and see if you're able to find the number in the middle of this circle here. Anyone have any thoughts? Take your best guess at what the number in the center would be. Well, if you've read your book, you probably have an idea of what the number is. But if you've never experienced what it's like for someone that is colorblind, what ends up happening is that the opsins are replaced. So in protanopia, where people have trouble seeing red, the red cones have been replaced with the green cone opsins. And there are a few different types of color blindness, but in essence, everything would look the same here. What I've done is used an app that I often use for making certain that figures in my papers and things like that are colorblind friendly. And I have taken one of these typical kind of colorblind dot tests, and I have turned it into a something where you can all kind of see what this would look like if we take one of the hues and we remove it. So in this case, the number that you would have seen would be the number 29. But when we're unable to extract that green information, all of a sudden, all of these blend together. We can also have this deuteranopia, where people have trouble seeing green. And again, what you would see is something that's very much a mosaic of dots that are now uniform instead of being able to see the green, because you would see these green components more like a red, with the green opsins having been replaced with red opsins. And tritanopia is probably the rarest, and this is where people lack blue in these cones. And even if there is some problem with these blue cones. Because of the way that this system works, there's more of an ability to account for these differences. So the reason why certain types of color blindness are more common is because at the level of the retinal ganglion cells, we end up developing and using an opponent color coding. So what we have are certain retinal ganglion cells that are excited when yellow is in the center, so they fire more action potentials, and they're inhibited by blue in the center. Right, and so the optimal would be if they have yellow in the center and blue in the periphery. That would be allow them to fire kind of maximally. And so this is exactly the same as we saw for contrast, but in this case it's for color. So we have another set where blue would turn the cell on in the center and yellow would turn it off. However, again, if there's blue in the surround, this cell is going to be partially inhibited by having blue off center. So this is this game where we're now able to detect color, but also contrast within color, because we have cells that are tuned very specifically to not only what the color is, with what their center field is receptive to, but they're also able to detect when there are stark differences between color from the center to the edge. So let's look at how this works in a practical example. If I were to have red light come in, then this is going to activate a red cone in the retina, and depending on what ganglion cell we look at, in this case a red on, green off, or a red center, green surround, this would activate this. So it would create an excitatory signal onto this ganglion cell. If we look at the case of green light, this is going to come in and it's going to activate a green cone, and on the case of the red-green ganglion cell, we're going to have an inhibitory input. So this is going to shut this cell down. And of course, the thing you should be thinking in your head is that there are also green-red cells where each of these actions would be the opposite. The other thing this should show you is that this is the reason why we can never really perceive a red-green mixture, because a red-green mixture, a reddish-green, it doesn't exist at the level of these ganglion cells. These are completely opposing responses that cancel each other out. In the case of blue light, we're going to activate one of these blue cones in the retina. And again, depending on what ganglion cell we look at, in this case a yellow on, blue off, we would end up with some kind of signal happening here. So in this case, the signal for blue would be off, which means an inhibitory signal. So this ganglion cell would be shut down. And part of the reason we choose to look at this one is because we should take a look at what happens then when we can blend colors. For example, in the case of yellow light, which we perceive as being a primary color. This is the point where it's kind of important that I go back and I point out the fact that these opsons aren't perfect. So I want to show you that the tail of what happens for kind of a blue cone and a red and green cone are in fact not perfect. So yellow ends up falling somewhere in the electromagnetic spectrum that's between green and red. And so what I want to point out is that these tails for all of these cones travel off in kind of different directions. So there's some overlap between these cones here. So what ends up happening then is that yellow light comes in and it activates the red and green cones equally. And this ends up doing something that's kind of interesting, because if we then look at what happens on that same red on, green off cone, that means that this is going to, the red component of the yellow is going to send an excitatory signal to here, but at the same time, the green cone sends an inhibitory signal. So these two effectively cancel out. However, what happens for this yellow on, blue off ganglion cell is that the signal from the red and the signal from the green both combine together and provide excitatory signals that activate this yellow ganglion cell. So the sum of these two is what ends up creating yellow on these yellow on, blue off ganglion cells. And that's part of why we perceive this as a primary color, because in this case, the two kind of partially active signals amplify onto this one ganglion cell. putting this back into our circuitry for a minute, one thing that's important to point out is that we talked last class about this idea that the processing in the lateral geniculate nucleus, or LGN, is very specific depending on the layer. And so what this means is that these ganglion cells, and of course the information coming through the cones vicariously, are traveling in a very specific path. So when we're talking about information for color, the magnocellular division, you can think of the M for magnocellular and M for movement. This is the main job of the magnocellular, if you remember, was movement. It doesn't respond to color, and yet the parvocellular and coniocellular each respond to color information. The parvocellular is particularly tuned to red and green cones, and the coniocellular particularly tuned to information coming through the blue cones. When it comes to sensitivity to contrast, on the other hand, the magnocellular division is getting a lot more information about contrast. And so this is more so coming from the rods and information about the kind of the center on for the light and center off cells. And the parvocellular and coniocellular division kind of poorly respond to that. Now, our blue cones are not as sensitive, and what this means is that the parvocellular, by getting red and green information, is the area where we get the highest amount of detail information in terms of what's coming into our visual field. So the parvocellular division is considered to be the area of the highest spatial resolution, but the color vision has all these computations that need to happen. So that happens through a lot of different processing, and because we have this one-to-one relationship between the cones and also these neurons that are projecting to the LGN, then this is a very slow response, whereas that amplification that happens at the rods, giving us information about contrast and light, happens very, very quickly in the magnocellular division. So we can detect changes much more rapidly there because of that convergence that happens with the rods down onto the ganglion cells. So just to give you kind of another map of this and to add a little bit more information, we have, in terms of information coming from both the ganglion cells and the cones, we have red and green information going to the parvocellular layer, blue to the coniocellular layer, and kind of contrast light-dark information to the magnocellular layer of LGN. And then I want to remind you that this information stays compartmentalized in the primary visual cortex. And what happens is as it goes to the secondary visual cortex, the dorsal stream is very sensitive to light-dark contrast and movement. That's probably the thing that it's most sensitive to. And the ventral stream is more sensitive to information about kind of color and form. So if you remember, the ventral stream is kind of the what. So we're processing information about what an object is and identifying it, whereas the dorsal stream is this where stream, telling us how things are moving through the environment. So this is not that these streams only receive this information, but this is where the kind of the bias of information is, where this magnocellular information is biased towards the dorsal stream, and the color information is biased towards the ventral stream, because that's what we need to get form versus movement. How do we perceive form? Well, we need to go back first in order to understand this, and we need to make a correction that I said we would make. So Hubel and Wiesel originally demonstrated that neurons are tuned to the orientation of lines. So we started to talk about these modules in the primary visual cortex, where one might respond to a line that's straight up and down, and another one might respond to a line that's horizontal instead. But what we found out in the time since then is that it's not quite exactly that simple. It's this idea that instead of responding to a certain line orientation, these neurons actually respond to a more abstract concept that we call spatial frequency. So it turns out that we need information about high and low frequencies so that we can understand what's going on in the world. And objects that have incredible amounts of detail tend to have really high spatial frequencies, while larger objects in our environment tend to have kind of lower spatial frequencies. And to be fair, the two are not mutually exclusive. So you're kind of seeing a low spatial frequency here as something that doesn't have much change across your visual field, and a high spatial frequency is something that changes quite a bit across your visual field. But what I want you to do for a second is I want you to take your left or right hand, whichever handedness you are, and I want you to hold up 4 fingers. And I want you to put them up in front of your face, very close to your eye. So put them maybe 2 inches from your nose. And so if you look at that, that's something at first that starts out with kind of a low spatial frequency. But if you take your hand and you move it away and put it now at arm's length, now all of a sudden, not only do you have a higher spatial frequency just for those same 4 fingers you're holding up, but you also start to resolve all kinds of other detail that you might not have been able to before when something was, pretty close. So you start to see the wrinkles on the back of your hand and all kinds of other high spatial frequency pieces. So there's something with these spatial frequencies that tell us more information than just level of detail. It tells us some things about, distance, how much of the visual field something takes up, and so on. So this concept becomes really important because this means that spatial frequencies change depending on us. It depends on how close we are to something. It changes depending on the viewing angle that we're looking at, all kinds of things like that. And this really comes down to a lot of differences in just how many cells are perceiving brightness at one time. So this is part of this bigger computation that the retina is going through. So one of the main things that spatial frequencies allows us to do is to decipher different types of information. And this can be a lot of things. It can be textures, or the way that things are moving, or any kind of pattern or spatial gradient. And if you look at the picture that you see here, I think that most of you, even if I were to show you just the left hand or just the right hand picture, would recognize this as a person's face. Some of you might even recognize that it's Abraham Lincoln. But in the one case, we have a lot of kind of high frequency information here on the left. And in the other case, it's been filtered out to remove it. Even though both pictures from an information technology perspective contain actually the same amount of information. So this shows you a little bit about just the differences in the way that our brain is kind of designed to process these different spatial frequencies. And so it turns out that that's what we now feel like neurons in the striate cortex, or the primary visual cortex, are designed to do. Now, we talked a little bit about neurons in the ventral stream yesterday, and this is what we think of as kind of the what stream. So we know that these primary visual cortex neurons are responsive to the features of different objects. And what we think happens is that this gets to the secondary visual cortex, and we start to combine these features together. So if you take information about color and about spatial frequency and you start to combine these together, then now you have information about a color and a pattern and how those are formed within the visual field. And then once this is in the secondary visual cortex, and has been processed all the way through it, we think the information leaving, at least based off of how people identify objects, is finally at the point where it represents an entire object, basically by taking the sum of all these features and sort of grouping them together. Now, one thing I want to point out here is that something like this is where we hit the limit of information. So we know what the brain does, but we don't have a perfect understanding exactly how it does it. We've now hit the point, unlike something like the action potential, where we know every detail from start to finish. We don't necessarily know exactly how this information is encoded, but we can figure out, by things like lesion studies or temporary inactivation in mice or rats or primates, that we can eliminate certain aspects of what they can perceive. So we can eliminate the ability to detect objects. And something really interesting that goes along with this is that if you look in different monkeys or different humans, we see that the basic spatial location of certain kinds of identification for what something is are grouped together. So if you look down at the legend down at the bottom here, and you look for where faces are represented, and you look at where this spot is in monkey J, and then you also look in monkey E, you see that, of course, first, both of them have a spot to identify faces, but the second thing that you should notice is that that spot is in pretty much the same location, and there is some drift. Probably the biggest drift I've seen in these maps is if we look for the location for where places are identified. That seems to be much more towards, I guess, what's this lateral edge in the one monkey than in the other. But nonetheless, there are similar features that are identified, and in somewhat similar parts of the brain for most of these. So, and it turns out that these can be grouped in very meaningful ways. So faces, body parts, objects, and places all have their different categorical organization. And this may have something to do with the way that these features are combined too, right? It may be that faces have a certain, more common spatial frequency that's a lower spatial frequency, whereas information about, objects may have some other features that the primary and secondary visual cortex use. And so this is a way that we should start thinking about this. Another thing that's really important to point out is that our ability to combine features is very dependent on learning. And so one of the things that can account for the differences you see across these brains that we just saw in the last image is the basic idea that not only do I have to recognize these patterns, but the way in which I learn these patterns could be very different across different individuals and also across different species. So if I'm trying to learn how to differentiate faces, some of you may be very good at this. You might meet somebody once, and you might be able to recognize them and identify them for forever and ever. And other people are practically face blind or are actually face blind and can never recognize somebody as many times as they meet them. And most people are probably somewhere in between. So a lot of this has to do with how we learn and how we integrate this information over time. As we saw in chapter 5, one of the ways that we can learn more about what the function of an area is are through lesions. And of course, we have lesion studies in animals, but in humans, we look for evidence of damage. And one thing that we've learned about in the visual system is that there are different kinds of agnosias where individuals might fail to recognize a face or an object or a scene. So one of the things that we've discovered is an area of the brain called the fusiform face area. And so if you look at the monkeys that had a specific grouping of activity for faces, it turns out that there's an area of the brain in humans we call the fusiform face area that recognizes these. And so if you were to show this picture here on the right to somebody with damage to that area, then they would see a lot of vegetables and flowers, but they would be unable to recognize that there's even the representation of a face embedded in them. We have agnosias that also prevent people from recognizing other things as well, objects, for example. So the fusiform face area falls into this ventral stream, part of the information about what's going on. And we see that it's activated in fMRI studies and things like that if people are viewing faces or if monkeys are viewing faces, much like we saw in that picture. And as I just said, damage prevents you from being able to effectively recognize faces. Another thing that goes along with the areas we just saw in terms of specific functional areas is the extrastriate body area. And this is also part of the what or ventral stream, or the what stream. And this can be activated by anything that kind of visually represents a body part. So, of course, this is actual body parts, but it can also be stick figures or anything else that approximates a body part. And one of the things that we didn't talk about so much, we talked about transcranial magnetic stimulation and the ability to induce magnetic fields, but one important thing to know about that is it can be either used to activate neurons or shut them down. And so if you disrupt this extrastriate body area by TMS, it turns out it's very close to the edge of the skull, so it's pretty easy to do this. Then you can cause people to not recognize body parts. And in fact, you can cause them to be unable to recognize their own arm and freak them out a little bit. And sometimes you might even see, pranks in a YouTube video where people wake somebody up and pretend that they've done something with an arm or a finger. As we kind of change consciousness states, we can also have a lot of trouble, correctly identifying what's our body and not, probably due to problems with the function for at least a period of time of the extrastriate body area. One of the ones I think is the coolest is the parahippocampal place area. So this is also part of information about what, but I think the thing that's interesting to me is that it's so very different, and it goes along with this area of the brain that we think of as being involved in many different kinds of memory, but it's activated specifically by scenes and backgrounds. But that's not the thing that I think is really cool about it. The thing that I think is cool is that looking at these things, it's not that scenes and backgrounds are comprised of the sum of all the different pieces. So it's not that, a room in a classroom, for example, is made up of the sum of, chairs and a blackboard and an instructor and a computer. It's actually that all of these are processed as kind of one giant context. So somehow the parahippocampal place area is able to group these things in a way that's different than when we look at the features of faces or specific objects. Moving on to the perception of location. One of the most important things for us to be able to perceive location is the fact that we have two eyes. So we have binocular or stereoscopic vision. And we also have the ability to make kind of, small adjustments. So we have ciliary muscles that control the lens of the eye. And by being able to both change focus and do this across two eyes, then we're able to use that information in different ways that we need to help perceive space. And this is something that you all do naturally every day, but you probably don't put much stock in what you're doing when it's happening. One of the most important features for us to be able to do this is, at least is surrounds an idea we call binocular disparity. So we've been talking to this point about how you should think about everything that your eye is seeing in terms of a grid, right? Where there's a certain set of cells on your retina that perceive how far to the left or right. You could think of this as an X-plane that something is, and how far up or down something is. Now, if you take this information and you add onto it the fact that we make specific calculations on top of this about where something is in the left eye compared to the right eye, then this starts to give us information about where things are in space. And this is incredibly important. So we have a number of neurons, and we were talking about these modules in the eye, and most of them, we said, respond to input from one eye or the other, right? So they can respond to input from either of the two eyes. But along with these neurons that form this really basic retinotopic map, where we just have this grid represented in space for different parts of the retina,
we also have a set of cells that we've found that are tuned to what you can think of as being retinal disparities. And so you should remember from last lecture that the idea of being tuned is that they fire more action potentials when a stimulus matches the features that they respond to. So in this case, some neurons respond when, let's say, something red is right in the middle, kind of in the middle of the two eyes, right? So it's hitting the foveas. However, there are other sets of neurons that will respond only when something is in the fovea of the left eye, but in the periphery of the right eye, for example. And so this is the idea of them being tuned to a retinal disparity. They respond most when the thing being perceived matches up in different parts of the eye. And this is part of the modules we use to detect where things are in space. So to just put this all back together, by having neurons that are tuned to retinal disparity, this means that these neurons are specifically designed to fire more action potentials when something is differently placed in one eye versus the other, which means that their job, at least in terms of how the brain is concerned, is to detect differences. And we're going to see some examples of this in the auditory system as well. It's a way that the brain seems to work out how to find things in space is by using almost an arithmetic calculation of the difference between one ear and the other, or one eye and the other. Two kind of interesting things about the location of objects too go along with the dorsal and the ventral stream. And so what we know is that damage to either the dorsal or the ventral stream will have a different function. So if we have damage to the dorsal stream about where things are, then as we're trying to perform actions that relate to where those objects are, we end up with trouble being able to perform those kind of actions. So if you try and reach to pick something up, then you have to do a lot of different things. You have to understand and get feedback from your eyes about where your hand is in space and what the size of the object is, and of course, how your hand relates to the object. And people who have damage to the dorsal stream have a lot of trouble being able to engage in those sort of spatial tasks. So much of what's going on in terms of visual location can be attributed to the dorsal stream. On the other hand, if we damage the ventral stream, then individuals can pick up objects correctly and use all of those important spatial features that we were just talking about. But the kind of interesting thing is they can do this and in some cases don't even recognize what the object is. So you could say, hey, can you pick up the object on the table, and they'll be able to pick it up and adjust the size of their hand to a mug versus a pen versus something else, and they can do a perfect job of picking them up, but not be able to tell you what any of the objects are. So even though you can't identify what the object is, you can perfectly identify where it is, and this is part of why we know these streams have different functions. And the last thing that we should really say about location is that an important job for the secondary visual cortex is to help process this information about disparities, but also to begin to send the information out so that it can be integrated with other cues. So the visual system doesn't act alone. Really, none of our sensory systems act alone. And we have what we think of as kind of like higher order association structures. This is part of where kind of the ending points of the dorsal and ventral stream head off to. So the integration of a lot of this information happens from projections that come out of the secondary visual cortex, especially to the parietal cortex, which is considered kind of a higher order association area. And without making it too complicated, because again, many of these things are kind of at the limits of our understanding in neuroscience, you should think of what's happening is that our body and our brain rather is beginning to put together a complete sensory picture of what's going on in and around our body. So then how do we perceive movement and orientation and things like this? Well, in some ways you all already know the answer to this. So let's put together a few things that we know. So first, neurons in the primary visual cortex. We've already said they respond to certain parts of the visual field. So our eyes know where an object is in the visual field. We've also said that these neurons are tuned to specific spatial frequencies, as well as a number of other things like contrast and edges and all kinds of things like that. And then the last thing that we just talked about is that the visual system can detect retinal disparities. So we have certain neurons that are tuned to when something is in a different place in one eye versus the other. So if you put this together, and this is maybe one of the more kind of profound ideas of this vision lecture, then you can imagine that spatial frequencies change as something moves. So let's go back to that example that I used once before. If you hold up 4 fingers 2 or 3 inches from your nose, and you move them back and forth this time. So move your hand completely out, extend it all the way out, and then bring it back to your nose, and do this over and over again. What's happening as something moves towards or away from you is that those spatial frequencies are changing. And in this case, if you had it perfectly aligned at the center of your visual field, you would never experience any kind of a retinal disparity. Your eyes would be seeing things equally at both times, but we can use spatial frequency in that case to discern that something is changing, right? Because the spatial frequency is being changed and updated. On the other hand, the retinal disparity, if you now hold your 4 fingers out at arm's length and you move something left to right, then the neurons that respond to those disparities now are going to be tuned in. So if you start out in the center and you move to the left, neurons responding to when something is closer to your left eye than your right eye are going to start being activated, and vice versa. And so in terms of left to right movement, or even up to down, then these neurons that are able to detect, retinal disparities are going to be able to do some of that job. And of course, part of what can happen with this, or part of what always happens with this, I should say, is that we have things moving around to different parts of the visual field. So even if you only had, one eye, if you were to cover one eye, like you often do in vision tests and things like that, and you take away the advantage of binocular vision, you would still have some idea of how things are moving. Now, you wouldn't do as good of a job with things like depth perception, but you would be able to detect, using all of these different combined features of the visual system, how something is moving. This concept is something that we call optic flow, and it involves changes in all kinds of different features of an object that give us hints as to how something is moving. So it could be a change in the size or the shape or the spatial frequency, or even our ability to perceive certain features of the object and observing them change as it moves through our environment. One of the more interesting kind of case study examples of damage is that there's an area of the visual system we call V5, and it's part of the secondary visual cortex. It's also sometimes known as area MT, and it contains neurons that are responsible for integrating all this information about optic flow. So they're not the ones processing the disparities or anything like that. They're the ones that are responsible for putting all of this together. And it turns out that in a very, very rare set of cases, there have been some humans that have bilateral damage to this area V5. And it produces this kind of a crazy disruption we call akinetopsia, which is an inability for them to perceive movement. And I think it's pretty hard to describe the way that these individuals go through their world, because it's as if the brain can take a bunch of still images without ever putting them together. So one of the examples that you can use for this is if you've ever seen a movie that's shot in any kind of like stop motion animation, which is becoming something that's less and less commonly used anyway as we gain the ability to do things with more computer-generated images. And so another, good example would be if you prevented yourself from opening your eyes until you stop moving. So if you were to try and navigate your world by closing your eyes as you walked forward, and you stop and you're allowed to get a picture, just one quick snapshot, you open your eyes, and then as you move forward again, you have to close them so you can't see any of the information as your location in space changes. Or if you were to try and navigate your world with just still photos by taking them on, your camera or your phone. Very interesting kind of disorder. The last thing that I want to leave you with is this visual summary that I pulled out of an old paper, and it does one of the very best jobs I've seen of just giving you a comprehensive view of the visual system, where we have cells in the retina, specifically the final output being these retinal ganglion cells that respond to different kinds of information. And this goes to the lateral geniculate nucleus, mainly the magnocellular and parvocellular layers, but don't forget coniocellular, not shown here, that have specific jobs like being color sensitive or color blind, for example, and that this information all comes into layer 4C of the primary visual cortex, and it remains segregated as it's being processed and is passed on to other layers where either movement or things like color and form are specifically processed. And also we have those modules for spatial orientation or frequency. And that this information is mostly passed on to the secondary visual cortex, where it starts out separate. You can see the 3 separate arrows coming in here, but it becomes more and more processed as it goes through the different layers of the secondary visual cortex, and is eventually passed on to kind of higher association cortices, or onto the different parts of the dorsal and ventral stream, where we turn it into places and locations, or even we process information about movement. And that's how everything is put together. So if you understand this diagram here, then I would say that you are doing a really spectacular job of having grasped all of the possible information that you might need for the quiz. And there's a lot of details that go along with this that are not shown here, but if you at least understand the flow of information here and are thinking about all the different things that go along with these areas, I would say you're at a very good starting point. So with that brings us to the end of chapter 6 on vision. And if you have any questions, now is a really good time to ask. Definitely doing it before you get into the quiz, or you can just shoot me an email with your question. If it's really broad, I might encourage you to pop into office hours or to schedule some office hours. So don't be afraid to use the emails or the office hours to get some help. It's a good time to do it. All right, take care and enjoy the rest of your week.